And then this is really just showing that again, right? The A zone is these really thick, dark areas because you have both the myosin and actin. The I zone is where you have only the actin, and then the middle would be the H zone, which is only where you have myosin. And this, again, this unit is called a sarcomere. So if we take a close look at actin at the molecular level, we find that there are two proteins bound to actin. One is called tropomyosin, and this is shown in these linear curved rods that are going through and attached to each band of actin. And then we also see this troponin complex, which is a protein that controls the positioning of the tropomyosin. So what I want to share at this point is that if myosin is down in the bottom over here, this cannot bind actin if tropomyosin is in between the actin and the myosin. So showing this here, we see the purple actin, and each one of those balls is a monomer of actin, and they're polymerized into a polymer of actin. Here we see myosin, and in order for muscle attraction to occur, myosin needs to physically attach to actin and then basically do a bending reaction to move and pull the myosin along the actin. And we'll take a look at that in a little bit. But I want to show you right now that we have myosin down here with this little crossbridge sticking out that can basically grab onto actin, but it can only do so if tropomyosin is not in the way. So when tropomyosin is in the way, myosin cannot bind actin. And here we see troponin, right? Troponin controls the positioning of tropomyosin. So the reason calcium is needed for skeletal muscle contraction is that when calcium binds to troponin, what it does is it pushes tropomyosin out of the way of myosin. So you can see here that the tropomyosin that was previously blocking myosin from binding actin has been moved by the troponin binding calcium. So basically it becomes activated. When this happens, what we can see is that the myosin can basically attach to the actin, and this is absolutely critical for muscle contraction. So muscle contraction is facilitated by sliding filaments. So previously I showed you that we have the A zone where the actin and myosin overlap, the H zone where we have myosin only, and the I zone where we have actin only. So during contraction, the myosin is going to grab onto the actin and then move it basically. So this whole sarcomere is going to shrink. Okay, so here's the extended sarcomere. It's getting smaller and then smaller. As the size of the sarcomere shrinks, what we see happening is the size of the A zones are increasing, and the size of the H zone is decreasing, and the size of the I zone is also decreasing. And then under the highest form of contraction, we see that indeed the A zone is going to be the dominant zone, taking up the majority of the sarcomere. So how does this happen? So once calcium binds to troponin, and that moves tropomyosin out of the way, allowing actin to be accessible to myosin, we need the myosin to have these heads that are binding to ATP. Now, when myosin head is bound to ATP, it is not in the conformation to be attaching to actin. So what needs to happen is that ATP needs to be hydrolyzed into ADP and phosphate, but they're still both attached to the myosin head. When this happens, the conformation of the myosin head changes. So instead of being kinked like this, it adopts a straight morphology. And when it adopts a straight morphology, we say the myosin head has been activated. And now it is binding again to the ADP and the phosphate, and now it can freely attach to the actin filament shown above. So a lot of the pictures I've been showing you so far depicted this configuration where you have the Z disc, right, the purple actin, the green myosin, and that's showing basically the side view. If we look at this is a lateral view. If we look at a longitudinal view, you can actually see these filaments. A side view would be like taking this slice over here and orienting it 90 degrees, so we're looking at it from this direction. And you can just see all the linear filaments inside the myofibril. So in order for the myosin to grab onto the actin and to induce contraction, again, we need the myosin head to be activated by taking ATP and hydrolyzing it to ADP and phosphate. And remember, both the ADP and phosphate remain attached to the activated myosin head. And this is critical for the myosin head to attach to the actin. So here we see the activated myosin head with ADP and phosphate attaching to the actin filament. Now, in order for the myosin head to be moving and inducing a contraction event, there needs to be another chemical reaction. And in this case, basically the phosphate, or in this chemistry, is going to be released. So basically the ADP stays attached. As the phosphate is released, that is going to be releasing energy. This is going to allow for the conformation of the myosin head to go from the extended conformation to the bent conformation. And in doing so, there's going to be a movement of the myosin, right? It's going to be moving to be, when it's straight, it's going to be moving to be, when it's bent, it's going to be moving myosin to the left, okay? So we can imagine that is moved this way with the power stroke. So a power stroke is the bending of the myosin head. This involves the release of remember that phosphate, which has a lot of energy, is going to be released, and then that has a conformational change in the myosin head. And it basically pulls the filament towards the center of the sarcomere. So after the power stroke, ADP will be released, and then a new ATP can bind. However, when the ATP binds, the myosin head needs to let go of the actin. Okay, so basically once ATP binds, the myosin head lets go of the actin, and then again, we need for the hydrolysis to occur, we need ATP to be hydrolyzed to ADP and phosphate. And once this happens, then the ADP and phosphate attached to the myosin head can now bind actin again and do another power stroke if needed. So the myosin head will move to the rest position, and it can rebind actin if the binding site is available, meaning we still have calcium binding to troponin to make the tropomyosin get out of the way. And sliding will continue until the sarcomere has been shortened up to the maximum, provided there's more signaling from the motor neuron secreting additional acetylcholine and setting this whole physiological process into play. So I'm going to review this one more time for you. So I remind you that we have ATP being bound to the myosin head, right? And when ATP is bound, it cannot bind to the actin filament, even if tropomyosin is out of the way. So what we need to have happen is we need ATP to be hydrolyzed to ADP and phosphate. Remember, both of these are going to remain attached to the myosin head, And when that happens during the hydrolysis of ATP to ADP and phosphate, we're going to see myosin head go from the bent conformation to a straight conformation. Then it is now activated to attach to the actin filament. And by the way, we call the actin filament a thin filament, and the myosin, we call it a thick filament, because myosin is actually more thick than actin for the filaments. Okay, so it's going to bind to the actin. We see that over here. It's attached to it now, as long as tropomyosin is out of the way, thanks to calcium being present. Then, in order for this to have a power stroke, we need for the phosphate to release. And as the phosphate releases from the myosin head, this will lead to a bending of the myosin head, right? So it's going to have a kink in it, meaning that this is now has movement, right? And after the myosin head becomes kinked or bent, the ADP is going to be released, as you see that little red molecule leaving, and then an ATP can come in, and once ATP comes in, that facilitates the detachment of the myosin head from the actin, and then the cycle can repeat if additional contraction is stimulated via acetylcholine. So what about if there's no additional stimulation with more acetylcholine? How does the muscle relax? So action potentials will cease in the T-tubules. Remember, the T-tubules allow for the action potential to be felt throughout the entire cell, because the whole cell needs to contract in order for the muscle contraction and to be facilitating the movement of a muscle. The calcium release channels will close on the sarcoplasmic reticulum. There are active transport calcium ATPase pumps that will move calcium back from the sarcoplasma to back inside of the sarcoplasmic reticulum. This is critical because we don't want the myosin grabbing onto the actin when there's no stimulus. So basically that tropomyosin will block that, and as long as there's no calcium binding troponin, the tropomyosin will be blocking the myosin-actin interaction. So that is really critical. And again, the tropomyosin will move back to the to blocking the myosin from binding the actin. So as you can imagine, we need tons of ATP for muscle contraction. So since muscle contraction can use more ATP than our cells can provide with cellular respiration, our body has a special mechanism to provide ATP under conditions where we need a lot of muscle contraction. So we have a molecule called phosphocreatine, and what we do is we have ADP combining with phosphate from phosphocreatine, and creatine is a molecule produced by the liver and kidneys, and what happens is when we're resting, phosphocreatine stores are replenished while we're resting, and then when we want to exercise, phosphocreatine can be a great source of additional ATP to facilitate muscle contraction. So here we have at rest, we had a meal, we have ATP, a lot of ATP from our metabolism, especially from oxidative phosphorylation. ATP can be giving one of the phosphates to creatine. That would convert ATP to ADP. It'll convert creatine to phosphocreatine. And then our skeletal muscle cells can be having a store of phosphocreatine, and then during excessive or strong exercise, what can happen is the phosphocreatine can donate as phosphate to ADP, creating creatine, and ATP. The ATP can facilitate muscle contraction, right? Because we need that for the power stroke of the myosin grabbing onto the actin and then physically pulling it. And we know from the during the power stroke, ATP is hydrolyzed to ADP and phosphate, and then their attachment to the myosin head controls the physiology of the myosin head.